Hello everyone, myself Gajanan Munde. I am here with my topic concerning about pre-PhD coursework seminar presentation. So my topic or a title of the topic is evaluation of some plant extracts for their in vitro nematicidal activity against plant parasitic nematodes which are associated with citrus species. So I'm from a zoology department, and the introduction of my topic, that is my work is on phytonematodes and their nematocidal activity associated with citrus plant species. So the citrus is the major and more important fruit crop in India and other countries also. So in other countries, near about 125 countries grown this citrus plant within the belt of 35-degree latitude and south and north of the equator. But the most citrus species is get attacked by nematodes, and that nematodes are more than one species. So the phytonematodes attack and causes some diseases like chlorosis, premature leaf drop, and stunting and dieback symptoms, which are seen in the particular citrus plants. The disease is becoming one of the most serious threat for the cultivation of citrus plants, and it will hamper citrus fruit yield. So it is directly a loss to the farmers and ultimately to the economy of a country. So the development of a chemical resistance has made to search for alternative ways for to control these nematodes, which are especially on a citrus species. So the phytonematodes become resistant to currently available nematicidal drugs, and some plant nematodes show the co-adaptation with the plants because of their evolution. And this is the major block in a treatment of nematode diseases related to this citrus species. So my idea for this particular topic is to develop of a chemical resistance has made to search for alternatives to control plant parasitic nematodes. Because actually these phytonematodes become resistant for these nematicidal drugs and so many treatments because of their co-adaptation with plants according to the evolution. But present study is an attempt to advise or devise various biocontrol measures which are based on herbal formulation, which are related to the natural one way. So this intervention will be developed for to promote the livelihood of poor agricultural farmers and are based on a cost benefit analysis of various disease control options. It will directly benefit it to that particular farmers. So the present investigation is aimed to study the in vitro nematicidal activity of some plant extracts which are naturally available on that particular nematodes which associated with that citrus species. So for this my topic, I use some materials and methods. So the some few steps are in this material methods. The first is a collection of infected plant material. So the preparation of a plant extracts, each plant material was thoroughly clean after collection with a water and dried under a shade for 6 to 7 days until it will be grindable or become grindable. So you have to grind it using an electric grinder, and then a cold extraction method is used, and also column chromatography will be for a plant material extraction. The isolation active column fractions will be processed for further phytonematicidal activity. Then the collection of a plant parasitic nematode species. For this present study, the nematode species collected from infected roots from the field of various citrus species which are available from Maharashtra state. So the collected phytonematodes are for preparation of a permanent slides and photography for a further study. Then the eggs and larvae of a nematode species will be isolated from infected roots of different species of citrus plants by using some methods like a sieve plant method and other slender methods. Then maintenance of the phytonematodes for in vitro testing. Then the infected roots were macerated in a 2% sodium hypochlorite solution for a 5 minute. To extract these eggs, and you have to get a centrifuge at a 1000 rpm for 4 minutes. Then the eggs were laid on a wet filter paper over water in pans for 3 to 4 days to hatch and the second and the third stage of a juvenile. So, that is a J1 and J3. So the in vitro nematicidal activity on phytonematodes, which are associated with our citrus plant. The nematode of the number of nematodes in an aqueous suspension was determined by using a counting dish. It is one type of a method. Then the collected nematodes will be studied group wise for a anti-nematicidal activity by using various active plant extracts. The activity was assessed by the determination of a time of paralysis and the time of immobilization or the death of the particular nematodes. For the purpose of this study, treatment will be replicated 3 times for to get better results. So the conclusion the conclusion of my this research work is to bioactive plant extracts offer a traditional alternative and also it is a natural alternative for a synthetic chemical phytonematicides and that are both sustainable and environmentally acceptable because of its naturality. Then the such plant extracts have a more important role in the future control of a plant nematode species. Then the present study will help coordinate and harmonize the national, regional, and global plans, and will help to improve programming and implementation of phytonematode prevention and control. So the outcome of this research has to potential to significantly alter citrus fruit production in a Maharashtra state. So it will alternately, finally, directly or indirectly benefited to the farmers and the growing economy of this country and other countries also. So this is my topic. So thank you so much.